Hello everyone. Today in this session I will discuss about how life start and the topic is origin of life. It is a part of your syllabus MB 4.1 and it is a part of evolution topic. At very first I'd like to discuss about a theory that is known as the abiogenesis or spontaneous generation. Abiogenesis is the generation of life from non-living matter. This theory states that complex living organisms are generated from decaying organic substances. For example, mice originated from stored grain, or maggots appear spontaneously in the meat. Francisco Redi, an Italian physician, was the first to disprove the abiogenesis theory in 1668. In his experiment, he took meat sample, heat it, and left the meat sample in a three flasks. One of the flasks is left open and the two others are closed. And he observed that the flask that is left open, there is a maggots, and the flasks that are closed, there is no maggots. So from here, he conclude that as because flies are able to come in contact with the meat pieces that is present in the unopened open flasks, so therefore there is a maggots. So he tried to conclude that the theory of abiogenesis is false. Later on, the John Needham in mid 1700 used an experiment to challenge Redi's work. He claimed spontaneous generation could occur under right condition. He heated chicken broth for a brief period of time and then covered the flasks, and after a few days small microscopic organisms developed in the flasks. So from this experiment had Needham proven Redi wrong. Later on, the Lazzaro Spallanzani, Italian naturalist, performed similar experiment to challenge Needham experiment. To do this, he took the nutrient broth, heat it, cool it, and left the flask open, and he observed there is a microbial growth. And in another set, he heat the flask of the nutrient broth, seal the flask, cool the flask, and then he observed there is no growth. When the flask is open, there is a growth of the microorganism. Spallanzani had proved that some microorganisms could survive in extreme heat, and more importantly, that Needham's theory of little animal, which is known as microorganism arising spontaneously, was false. Unfortunately, many scientists were not convinced by his experiment. Finally, Louis Pasteur, a French chemist, biologist, and microbiologist, in his famous swan neck flask experiment, finally disproved the theory of abiogenesis. In his swan neck flask experiment, he took the nutrient broth in a swan neck flask, heat it, cool it, and left the flask sit, and he found that no bacteria present. In another set, he hit the flask, broke the swan neck, and he observed there is a microbial growth. And in another set, he hit the flask, tilled the flask for some time, and he observed there is a microbial growth. So from here he conclude that when the flask, swan neck flask is remain intact, only air can enter without microorganism, so therefore there is no bacterial growth. But when the swan neck is broken and the nutrient broth were come in contact with the microorganism that is present in the air, therefore there is a microbial growth. So from here a new theory of biogenesis came, and according to this theory, living things come from living things. For example, mice come only from mice, and microorganisms can only from other bacteria. Then how does life start on Earth? Since spontaneous generation was proved incorrect, many scientists began to wonder how life started on Earth. The Earth formed about 4.5 billion years ago. Life's origin began at least 3.5 billion years ago. A.I. Oparin, a scientist, attempted to answer this question. The spontaneous generation debate heated up again. A.I. Oparin, a Russian biochemist in 1924, proposed that life had arisen from simpler molecules on lifeless Earth under much different atmospheric conditions than exist today. Life originated in a very long period of time. Oparin and Haldane proposed a theoretical model of chemical evolution. This model suggests that under the strong reducing condition which is prevalent in the early earth, because early earth is a reduced environment, early earth has had reduced environment, inorganic chemicals would spontaneously form organic molecule, simple sugar and amino acids. In 1929, J.B.S. Haldane proposed that UV light acting on the primitive reducing atmosphere containing water, ammonia, and methane produce oceans with the consistency of hot dilute soup containing building blocks of life. In 1953, Stanley Miller and Harold Urey tested the hypothesis of Oparin-Haldane by constructing an apparatus that simulated Oparin-Haldane early Earth. Methane, hydrogen, ammonia gases, which are prevalent in the early Earth, were introduced into a moist environment above a water containing flux. This system is supplied with electrical current to simulate the primitive lightning discharge, and they finally found that the presence of amino acids like glycine, alanine, aspartic acid, glutamic acid, hydroxy acid, urea, lactic acid, glycolic acid, succinic acid, propanonic acid, etc., biomolecules or organic molecules were being formed. So simple biomolecules that are formed were further polymerized to form polymeric macromolecules like polysaccharide, polypeptide, etc. So from here the concept of proteinoids came. Sidney Fox and co-worker produced synthetic peptide-like product of molecular mass between 4 to 10 kilodalton by heating amino acid mixture at 150 to 180 degrees centigrade, which are called proteinoids. Unlike linear protein produced in the cell, proteinoid are branch polypeptide chains. The second concept of quasarvates also taken into consideration. Quasarvates is an aggregate of colloidal particle due to acidity, temperature, etc. in the liquid phase that persist for a period of time as suspended membrane droplet. Another concept of microspheres are also taken into consideration. When proteinoids are boiled in water and allowed to cool, a microscopic membrane-bound spheres or microspheres are formed. These quasarvates and microspheres indicate the formation of primitive cell-like structure called protocell. And more importantly, due to the amphipathic nature means a molecule which has a hydrophobic group and as well as the hydrophilic group. So that's why they behave distinctly into the water. So the lipid molecule, this amphipathic nature of the lipid molecules, they are able to form the membrane in the water and in the primitive times. So what is the possible mechanism of evolution of life in this earth? At first the earth is sterile. The first molecule that is being polymerized, which has a signature of the life sequences, that is known as the RNA, according to the RNA world hypothesis, because RNA has a capability of self catalysis as well as self replicating. So that's why the RNA molecule can produce its own copy of another RNA. Gradually, this RNA molecule is entrapped into the lipid vesicles, which is known as the early cell, and in this lipid vesicle, some protein molecules are entrapped, and later on, the catalytic function is taken over by the protein, and RNA is only having the function of replication. And later on, gradually, the RNA replication machinery of the RNA is replaced by the DNA, and the central dogma established of the modern cellular life, that is DNA from RNA and RNA from protein. So now the most important question, what is evolution? A gradual process in which something changes into different and usually more complex or better form is known as evolution. An usual misconception that evolution is a theory of the origin of life. No, evolution is not the theory about the origin of life. Evolution is the theory which deals mainly with the fact that is how life become modified after its origin. Okay, so in the next session I will continue with the remaining portion of the topic. Thank you.